Hey Beaks, it's Mr. Boylan. Today I'd like to talk to you about DNA replication. So we are in S phase of the cell cycle today, right? Okay, here we go. So 1953, James Watson and Francis Crick, they introduced this elegant helical DNA double helix molecule, and they were the first people ever to come up with an accurate model, and we'll see why theirs was so accurate. They had a lot of help from a lot of different people, including somebody who wasn't treated very fairly, which was Rosalind Franklin, who took the famous Photo 51, right? So DNA is probably the most celebrated molecule of our time, right? Because this is our genetics. We just got finished learning all about genetics and all the crazy patterns and things that happen there. Well, hereditary information is encoded, right, by DNA in all your body cells. And this DNA is kind of like the blueprint for life, right? It's like a hard drive inside of there that directs all of your biochemical, anatomical, physiological, and behavioral traits. So very important. Here's a quick video for you.
The structure of nucleic acids, DNA and RNA. There are two main types of nucleic acids, DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. Nucleic acids are large molecules made up of smaller molecules called nucleotides. The nucleotides in these molecules are linked together through covalent bonds, or bonds where electrons are shared between atoms. Let's start by looking at DNA. The nucleotides that make up DNA have three parts: a phosphate group, a nitrogenous base, and a deoxyribose sugar, or 5-carbon sugar. The phosphate group consists of a phosphate bonded to 4 oxygen atoms, with one of the oxygens bonded to the number 5 carbon of the deoxyribose sugar. An important note here: the number 2 carbon of the deoxyribose sugar is bonded to a hydrogen atom. Carbon atom number 1 of the deoxyribose sugar is bonded to the nitrogenous base. There are 4 types of nitrogenous bases that can be found in DNA. Adenine, shown here, guanine, shown here, cytosine, shown here, and thymine, shown here. As you can see, adenine and guanine have double ring structures, and cytosine and thymine have single ring structures. Adenine and guanine are called purine bases, and cytosine and thymine are called pyrimidine bases. DNA is a double-stranded nucleic acid, and its molecules take on a helical formation. Each helical chain has its phosphate sugar group toward the outside, and the nitrogenous bases facing inwards towards the nitrogenous bases of the other chain. Each of the bases on one chain is joined to the base of the other chain through either 2 or 3 hydrogen bonds. Thymine and adenine are joined by 2 hydrogen bonds, and cytosine and guanine are joined by 3 hydrogen bonds. Thymine and adenine are always a base pair, and cytosine and guanine are always a base pair. It's estimated that DNA molecules contain more than 100 million of these base pairings, and in one individual's body, the sequence of the base pairings is the same in every DNA molecule. So this sequence of base pairing is unique to that individual. DNA is often called the information molecule because it contains the master code needed to make various RNA molecules and protein molecules in the body. Now for RNA. The nucleotides that make up RNA are very similar to those that make up DNA. RNA nucleotides have a phosphate group with the same structure as in DNA. They have a 5-carbon sugar, but the number 2 carbon is bonded to a hydroxyl OH group instead of a lone hydrogen atom, and this sugar is called ribose. It also consists of the 3 same nitrogenous bases as DNA: cytosine, adenine, and guanine. But it does not contain thymine. Instead, it contains uracil, which is a single ring structure like thymine, making it a pyrimidine base. Most RNA molecules are single-stranded nucleic acids, and many times they form a folded, compacted structure with some hydrogen bonding taking place within the base pairs of the molecule. These pairings are the same as in DNA. Cytosine and guanine are a base pair, and uracil takes the place of thymine and pairs with adenine. RNA is extremely important in the process of forming different proteins in the body, of which we will cover in depth in a later video. And that be the basics on the structure of DNA and RNA.
Okay, so you can put down your popcorn now, and here we go, we continue. So there you can see, and like the video just said, we have some important things here. These sugars in here, this is deoxyribose because this is DNA. The carbons are labeled 1 here, 2, 3, 4, and then 5 hangs up here off the chain. That's why this is called the 5' and we'll get into this again later. And as you can see, it's antiparallel. So the 5' is here, that means 3' is here, and it's antiparallel, so 5' here and 3' here. Okay, so I know a lot of that was a review, but just make sure you study the structure again of DNA to help you out. So like I said, we're in S phase here, and the relationship between structure and function is manifest in the double helix, right? It has a very specific shape, and because of this, the base pairing happens and enzymes can work really, really well, whether it's making new strands of nucleotides or whether it's repairing strands that we already have. So, since the two strands of DNA are complementary, each one acts like a template, right? So here's our double helix right here, but when it separates, it acts almost like a magnet here, like a template. It wants to join again. So this adenine would love to have a thymine come and join, and this thymine would love to have an adenine to come and join. And that's how you end up with two identical molecules here. And if you're good, you know what these two things are actually, and you would be sister chromatids, right? We made our sister chromatids here. Okay. Now, Watson and Crick's model is what we call semi-conservative, right? There is other theories out there on how DNA copied itself, okay, but semi-conservative is the way that we do it, meaning, sure, we use it, okay, but we conserve somewhat of our DNA, right? Each new strand is going to have half of the old strand. So we conserve half of it, so it's semi-conservative, right? As you can see here, half old, half new is what we're making. All right, and here you can see the different types of models that people were thinking about. Okay, and I'll show you how scientists discovered this semi-conservative model was correct. Okay, another quick video for you here.
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles, which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
Okay, so moving right along here, we have Meselson and Stahl, and they came up with a really cool experiment to figure out this semi-conservative model. And I'll be honest with you, some people don't get this right away, and you might have to actually draw it out to really understand it. They used radioactive nitrogen in their experiment, but let's take a quick look at it. But again, you might need to pause the video, go back, read the textbook again, and really try and figure this experiment out. So, it's a nice picture, isn't it? It's a beautiful sunny day, sitting on the grass, eating sandwiches, whatever they're doing. Pretty nice. So, here you can see that we have bacteria cultured in a heavy nitrogen right here. Oh, I'm sorry. So the bacteria are grown in a heavy isotope of nitrogen, nitrogen 15, right? So let's just consider all the DNA that's coming together here, the nucleotides, because remember, nucleotides have a nitrogenous base, so that nitrogenous base is going to be heavy, dark blue. But then they're going to transfer it over here to where we have the lighter isotope of nitrogen, nitrogen 14, more common, right? So what's going to happen here? Well, they're going to let the bacteria replicate. And if you think about what's going to happen here, we're going to end up with a single band, right? Because what's going to happen is the dark blue DNA is going to rip open, and what's going to happen is light blue or lighter nucleotides are going to join on to those template strands. So basically all the DNA right here after the first round, this is like a gel where you separate things based on density. When you have this one band here, it means that, oh, we have right here, we have the old strand opened up and the new strand is lighter that joined to it. Okay, so old and new is connected right to it. So we kind of got rid of a conservative model right away because we didn't have any heavy dark blue here. Okay, so now take a look though after a second round of replication. So what's going to happen here is, well, we're still going to have our light blue, dark blue in the middle, but think about what's going to happen there. When we rip apart the light blue, dark blue, the dark blue is still going to be, well, filled up with light blue because we're inside of this flask right now. Right? So you're still going to have that same layer right here of light blue, dark blue, but then what's going to happen is, what about the light strand? The light strand that is now used as a template will fill up with light nucleotides in here, and that's why you get this brand new strand here, right? And that actually negates the dispersive model. Now we know that, oh no, it actually is semi-conservative. These guys are pretty sharp, right? And what's going to happen is, you can see here, the lighter band is going to keep getting bigger and bigger and bigger and bigger. Right, so here's after one replication, 2, 3, 4. So like I said, if the semi-conservative model experiment doesn't make sense to you, you might need to actually just pause the video, take a minute, go back and read it, and try and make sense out of that. Okay, so let's take a closer look at replication. It is super fast and it's super accurate, but mistakes of course do happen. And there are more than a dozen enzymes. We're just going to learn a couple today. Okay. Before continuing, like I said, it's very important that you understand the 5' and 3' ends of DNA. So here's 1/2 of DNA right here. Okay, the H bonds will be forming here to the other side. But see the sugar here? 1, 2, 3, 4, 5 carbons. And because of those carbons, where you have 1 is always connected to the base. Okay, 2, I can tell this is deoxyribose because there's no oxygen there on the 2 carbon. 3 always bonds to, this is where you're going to form your phosphodiester bond to the next phosphate group or the next nucleotide. 4, and then 5 is where you connect your P group to. Okay, so this nucleotide right here, this is one nucleotide. It's very important to understand the 1, 2, 3, 4, 5 prime ends. Okay, or the 1, 2, 3, 4, 5 sugars, the carbons in the sugar. Okay. Okay, so replication begins at particular sites called origins of replication, and this is where you're going to open up, okay, the DNA double helix, and this is what forms this bubble to happen. Okay, now a eukaryotic chromosome can have hundreds or thousands of origins of replication, and that's super important that we have that because this would take forever if we didn't have a bunch of origins. And replication proceeds in both directions. Now, this is a little confusing, and I don't know if a video is the best way to teach this because, think about it, this is going to be what we call the leading strand, and this is going to be the leading strand, but then we're going to have lagging, 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 lagging. And what does that show? All the arrows, depending on which strand you're on, okay, either the top strand or the bottom strand, all the arrows go in the same direction. Okay, and that's because the enzymes that do the work here can only work in one direction, because as you know, enzymes are very specific. So, grab your popcorn.
Replication of a DNA molecule begins at a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules.
Okay, so that video actually wasn't great except to show how the bubbles come together, because that was showing how DNA grows out in both directions like this. That's not true. From the origin of replication, you're only going in one direction. Okay, remember all those arrows only go in one direction, whether you're on the top or the bottom strand. So, but the bubbles do join. And as you can see here, look at if you're a prokaryotic cell, right? These guys over here, they just have, in order to replication, they just have to copy a circle, basically. But you have linear pieces of DNA, and we have multiple origins, and multiple bubbles will happen, and then we'll get our sister chromatids here, right? And if you forget what that is, don't forget, by copying your DNA in S phase, when we go to cell division, you don't just have one strand of DNA, you have two, right? And these are your sister chromatids that are held together at the centromere. So it's a good thing that we do this replication the way we do, so we could divide our cells. Okay, so there's lots and lots of enzymes here. At the end of each replication bubble is a replication fork. Okay, so we call this area here the replication fork. Now, what's happening here is lots of things, actually. Okay, I'm just going to go over what some of these enzymes do. First of all, you need a primer here. You need an RNA primer, and I'll show you why in just one second. But you have to actually lay down RNA before DNA, and that's actually really cool that we do that. It's probably because RNA was on our planet first, okay, before DNA, but we'll talk more about that in a second. You also have these single-stranded binding proteins because helicase is sitting here. And what helicase does is helicase breaks the hydrogen bonds, basically, between the double helix here. But the thing is, if you're going to break hydrogen bonds in DNA, right, it wants to come right back together. Remember that it's like a template. So this guy still wants to bond with this guy. They want to come right back together. So by adding some single-stranded binding proteins, the DNA won't re-anneal. It won't come back together. Okay, so helicase is going to unzip the double helix. Single-stranded binding proteins prevent it from coming back together. RNA primase is how we begin making a new chain. Even though it's RNA, we'll get rid of that later. And then you have this guy out here, topoisomerase. He's very important because you don't want the chain to be overwound. Okay, it could actually tighten too much. If you think about playing with strings or something like that, if you keep wrapping strings together, and pulling on them, and they're all coiled up, it'll get super tight and could destroy it, right? So topoisomerase kind of keeps breaking and releasing the pressure of what we're doing here with replication. Okay, so there you go. The single-stranded stabilize it, and topoisomerase prevents overwinding. Okay. DNA's polymerases cannot initiate the synthesis of a polynucleotide. They can only add nucleotides to the 3' end. So what's going to be very important is 5 to 3. 5 to 3. Almost everything we talk about with this chapter is going to be 5 to 3. So if you have 5 to 3, talking about the carbons in the sugar, you're going to latch on to the 3' end. It can only add nucleotides to the 3' end. So let's pretend my pinky is the 3' end and my thumb is the 5'. Well, there we go. There we go. There we go. You can only go that way, right? So what is up with this primer? Well, let's see here. Why do we need primase? Why do we need to add an RNA primer right here? Give me 1 second, I'll show you. Okay, so I'm back. And you probably know what these things are here, right? These are little Lego Duplos, okay, that my daughters like to play with when they were younger. And I want to tell you about DNA polymerase. Okay, he's not the brightest enzyme, because what happens is when we want to make a new chain of DNA, you would think DNA, polymer, many units, right? ACE enzyme, DNA polymerase is the main guy, which it is. The problem is DNA polymerase, in this case DNA polymerase 3, he's not too bright. He doesn't know how to take these Lego Duplos and put them together. But there is a guy who does know how to do it, and his name is primase. Right? RNA has been building RNA nucleotides together for a long time, or RNA primase. We've been making RNA since probably before life existed on this planet, which is crazy. So what happens is primase says, oh, I see, let's just pretend the bottom is the 5' and these little holes up here, they're the 3' end, right? So RNA primase, okay, can go, sure, let's connect. Let's connect. Oh, this is what we're up to. And all of a sudden, okay, DNA polymerase might look at this and say, wait a minute, I know what that is. I see a chain of nucleotides. And then DNA polymerase can jump in here and say, hey, okay, I'll take over if you don't mind. Sure, here I come. And DNA polymerase can now start adding nucleotides, okay, to the RNA strand, and now we're adding DNA. Oh, there we go. So DNA polymerase might not be smart enough to start it from scratch, but once he sees a chain and he sees that juicy 3' end hanging off the end, then he can jump in, okay, and start adding the right nucleotides. It's kind of the same thing that happened with my daughter when she was young. When my daughter was young, I gave her a couple of these, and she was acting all like DNA polymerase. If I just gave her a handful of these, she sat there and she looked at them, and right, she didn't know what to do with them. But then I gave her this. I gave her a stack of them, and then I gave her a couple extra, and she said, when she put it together, I was so proud of her, right? Because just like DNA polymerase, she needed to be taught. She needed to be shown first, okay, what to do. I'm sure that was amazingly helpful for you. Here's hoping. All right, let's go back here. So, we have an enzyme called primase, like we said, starts it. And the primer is short, 5 to 10 nucleotides long. But that 3' end, that's going to call in DNA polymerase. So these enzymes, there's different DNA polymerases, and they do slightly different jobs, all of them. They catalyze the elongation of new DNA at the replication fork, okay, or we'll see some other places. So most require a primer and a DNA template strand, and bacteria do this a little faster than we do, but that's okay because we have a whole bunch of origins of replication, like we said, so we get it done fast as well. Now this is antiparallel elongation, okay, and it does affect how we do replication here. All right, because DNA polymerases only add to the free 3' end, right? You're always going 5' to 3'. You can only go in that direction ever. All right, so if you look at this picture here, we put a primer on. Okay, here's our primer, and it was heading this way. And there you can see we have this guy called the sliding clamp. That's just like it's called a processivity factor. It's just helping this all happen correctly. But DNA polymerase, he's going to start adding the right bases now. But see how the arrow's going this way? And let's fast forward a little while. See how we go this way and look at all this time? You know what we didn't do? We didn't do the bottom strand yet, because the bottom strand would have to go in the opposite direction. Okay, so we're going to have something called the leading and lagging strand here. Back to this, right? So what I just showed you there was the leading strand. Here's all those enzymes, and here's the replication fork. But the lagging strand is going to have to keep playing catch up, catch up, catch up. And if you watch this video enough times or read the book, or if you truly understand this, you're going to understand that there's no such thing as a leading strand on the top and a lagging strand on the bottom for the whole molecule, right? Wherever your origin is, you're always going to have leading strands going in opposite directions. So the bottom and the top is a leading strand. But behind the origin, you have to keep playing catch up, right? So if you notice what I mean there, take a look. You have the origin of replication, leading strand, put a little primer, leading strand, put a little primer, leading strand. But now, because you can only add to the 5', 3' direction, and DNA is antiparallel, you have to keep playing catch up. These are called lagging strands. You just keep going. And you can't make these lagging strands until the opposing leading strand goes far enough to open up more DNA so you could join on. Right? It's very confusing. Okay? And it takes kids sometimes quite a few times of reading the textbook, going over videos, looking at it over and over and over again till they get this. So, again, in this case, we have a lagging strand. Okay? So, I don't see the origin of replication around here, but we're still going to go ahead and put on an RNA primer. And then DNA polymerase says, oh, look at that. I can jump on here now. And it starts going along. And that polymerase will keep moving until it hits another RNA primer. And it's going to keep moving along. And eventually we're going to have another special enzyme that's going to close gaps. This one, it's going to remove the RNA. Okay, it's a different DNA polymerase. It's going to knock out the RNA, and it's going to put in DNA. So now, look at that. We have a strand of just DNA. And now we have a little gap in there, so we're going to have another enzyme to fill the gap called ligase. Okay. So what do you call these fragments as you're making them here? These are Okazaki fragments. Right, so I'm making a fragment here, and eventually there'll be another fragment coming in, connecting to this fragment. These are Okazaki fragments. So eventually you have to get rid of the RNA, and ligase will connect the fragments. Okay. So I do like this picture quite a bit. All right, as you can see here, lots going on. Okay, so do you have to be an expert on all these enzymes? No, but if you know them, that helps quite a bit. So let's take a look here. We had the origin of replication, okay, somewhere over that we barely can see now, okay, on this picture. And here's the leading strand. We're going to just keep going. The leading strand is continuous, right? So we have DNA polymerase 3 that's doing its thing. It's going crazy. Helicase keeps unzipping the double helix. Single-stranded binding proteins aren't allowing the DNA to come back together. Okay. And then down here, we've been doing the lagging strand. Notice this one has to go in this direction. The top was leading and continuously built this way. The bottom is lagging and has to be built this way. But because you can only go in these opposite directions, the bottom has to wait until the top gets further along. So in other words, this strand, sure, is already made, and now this strand is made, but until DNA polymerase 3 up top went through here, that's what opened up enough space here, so you put on another primer, and then you'll add some more DNA at the bottom. Right, and that's going on both sides of the bubble, okay, both forks. Pretty tricky. Now, that's hard enough to understand, but actually the way this really works is you have a whole complex DNA replication machine going on inside your cells, right? And all of this stuff is held together by proteins, and it's absolutely mind-blowing how this all works together. And I mean, it had, what, billions of years to get this just right. We're still not just right? But we're pretty good. So these enzymes and proteins and everything all work together inside of your cell. So this is all going on during, like we said, the cell cycle during S phase. Okay, and parts of it may be, you have like stabilizing connecting proteins in here, so it may be held pretty stationary. And you'll see in the BioFlix movie eventually that I'll show you that the DNA polymerase molecules almost seem to reel in parental DNA and extrude newly made daughter molecules. It just kind of knows how to pull it all together just right, so you can keep the leading strand going and pull in the lagging strand. Very, very cool stuff. Okay, so this one's a little better. Here we go. If it'll open up for me. There we go. Hear that music? That's amazing.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from reannealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase 3. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase 3 lays down new DNA. The process repeats again and again. DNA polymerase 1 replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragment.
Okay, so I paid a lot of money for that segment there. Not really. As we go through here, okay, so this is the BioFlix, and I'm not going to go ahead and show this online, copyright reasons and stuff, but I will be sharing this with my students. Okay, so, and that's really going to help you understand what's happening here. Okay, seeing the BioFlix, and that shows the whole machinery at once. Now, last part for today, there are some issues that happen here, right? You do make mistakes sometimes, and if you don't catch these mistakes, that's a mutation, right? And some of them, as you know, can be heritable. But for the most part, you're mutating somatic cells, no big deal. Well, it could be a huge deal, but you don't pass those on. So DNA polymerases can also proofread newly made DNA, replacing any incorrect nucleotides. Mismatch repair of DNA, repair enzymes correct errors in base pairing, pretty cool. You have genes that can actually make these enzymes, which is pretty neat. DNA can be damaged, lots of different reasons. Usually when I'm teaching this unit, it's around prom season, and at Pennsbury we have the greatest prom in America. So although this year we're stuck in with the coronavirus, so I will see what happens. But I always warn kids. I say, oh my gosh, what are you doing going out in tanning beds and things like that? Why subject yourself to these harmful, high radiation machines when you know it damages your DNA? You wouldn't sit there and smoke cigarettes, I would hope, right? You don't get x-rays every day, so You have to be very, very, very careful of these harmful rays, because DNA can get damaged, and if it does, it can make changes. It can also just happen spontaneously if we're unlucky as well. So, nucleotide excision repair, a nuclease cuts out and replaces damaged sections of DNA. So, for example, over here you can see, all right, we have a problem, and we have, oh, here we go. This would be an endonuclease that cuts out the bad part, and DNA polymerase fixes it, and then ligase comes in and seals the deal, right? Creates those phosphodiester covalent bonds again. Now, error rate after proofreading repair is low but not zero. And why do I have Chucky D here, Charles Darwin? Because thank goodness it's not zero, right? The reason that we're not amoebas right now, and the reason that we're these complex human beings, is because of mutations that have happened over a long time. So sequence changes may become permanent, can be passed on to the next generation. Sometimes mutations can be beneficial. Okay, they're the source of genetic variation on which all natural selection operates. So we've already talked about meiosis, right, and sexually reproducing organisms, that you have the crossing over and the independent assortment, right, random fertilization. But what's happening here is, no, we're just talking mutations. Mutations are the raw fuel, the substance of what evolution is based on. Okay, so it's very important that we have mutations happen. Although sometimes, of course, it can be sad if they happen at the wrong spot. Could really cause some problems. Okay, and the last part here, replicating the ends of DNA molecules, and very similarly to the when we talked before about Meselson and Stahl, that's tough for some kids with the radioactive nitrogen. You might have to go through this a couple times until it makes sense to you. But once you understand replication, this should click for you. Okay, but what's happening here is polymerase, as you know, is limited for a couple different reasons. But we have linear chromosomes, right? So if you think about it, polymerase might be able to copy continuously on the leading strand, but eventually the lagging strand at the very end of the chromosome might not be able to catch up. And you might actually have a chunk of DNA that doesn't have a partner, basically. And what can happen then is you cut it, okay? So technically, you're going to be shortening and shortening and shortening your chromosomes on every replication event, okay? And you might be thinking, well, that's terrible, but no, that's normal, right? We'll see. So this is a prokaryotic chromosome, and as you can see, oh, this is not a problem, okay? This is a circle. So, well, this is not a circle. This is obviously some sort of wolf bunny, right? There's the tail and there's the head. But point is, yeah, they have a circular chromosome, so they don't have to worry about ends, right? But for us, oh yeah, we have trouble here, okay? Because what's happening is, well, the leading strand, okay, is just going to get continuously made, but here we're on the lagging strand, okay? So if the polymerase and everything goes this way, follow my cursor here. Well, what if we never put a primer here and can do this last little part here? If we don't lay down a primer here, we can't fill this gap and we cut it loose. And we'll have shorter and shorter and shorter and shorter and shorter chromosomes. That means every time we do S phase or replicate our DNA. And that's exactly what happens, right? But that's okay, because we're made to do that. The ends of our chromosomes are what we call telomeres, okay, or telomeric DNA. So eukaryotic chromosomes, excuse me, have special nucleotide sequences at their end called telomeres. And they do not prevent the shortening of DNA molecules, but they basically prevent or postpone the erosion of genes. In other words, you don't have anything too important, per se, at the very ends of your chromosomes. And by the time you start getting into those genes, you've shortened your chromosomes that much that you're actually starting, you're probably very old, and you probably have a lot of other stuff going on at that point too. So possibly, right? So what's going on here is we have kind of a cushion. It's kind of like a bumper on a car. So when you mess with a car, hey, if the bumper, okay, if you get into an accident or something, if that's the thing taking the damage, your car is still okay. Your genes are still okay in this case, okay? A lot of people do think that telomere shortening triggers aging, or it has something to do with aging. Possibly. Okay, that could be true. But there's something interesting here. You have to, divide your chromosomes many, many times including when you're making germ cells. Well, that's a problem, right? Because we have to make sperm and eggs for our kids. So that's not good. Are we going to be giving our kids shorter and shorter chromosomes every time? No. Okay, we have an enzyme called telomerase. And what telomerase does is it extends the telomeres. So, okay, as long as we have telomerase active inside of a cell, we're going to, every time we divide, we'll go ahead and extend those telomeres all over again. And some people, maybe you right now, are thinking, ooh, is that like a fountain of youth? Perhaps we can negate aging if we just keep lengthening our chromosomes. And it's kind of a weird and debatable topic right now, right? Because your cells have a programmed number of cell divisions, right? Death is a part of being a cell. So when you reach the point of so many divisions, your cell should die. Okay, so extending the telomeres might not be in our best interest in certain cases. So it's a lot of people are talking about this. Here you can see this another picture of what's going on here. So telomerase is going to go in here and say, hey, I'm going to lengthen the lagging strand for you, and then DNA polymerase is going to come in and say, oh, thank you, I see a juicy 3' end here, and it's going to join on here, and you're just going to keep lengthening the end of chromosomes over and over if you have telomerase there. And actually, there is a type of cell that does this besides your germ cells, and we've seen this in cancer, right? And if you think about it, cancer's immortal rule-breaking ability is because as these cells keep dividing and growing and dividing and growing, why don't they stop? Why don't they die? Because aren't you supposed to when you get short enough? And the problem is, they keep extending their chromosomes. A lot of times cancer cells have this telomerase activated, so they keep lengthening. So we can't beat them that way. Our built-in safety mechanism, I guess, against it, right, gets defeated because they keep lengthening the chromosomes. And that can exacerbate the disease, which is sad. So maybe if we can find a way to shut down the telomerase there, that would help us. Who knows? Okay, so that was a lot. Okay, that's replication, and I'm going to show you a couple pictures. Okay, very quickly that you can look at, and I'm going to share these with my students, so you can look at these again and just try to wrap your head around it. But if I had to say what's the main thing that kids struggle with, it's the 5', 3', and it's the leading and lagging. Once you wrap your head around that, easy stuff. The Meselson and Stahl experiment, no problem. The telomerase and telomeres and all that, no problem. Okay, semiconservative, no problem. But you have to get down the 5', 3' leading and lagging strand first. Okay, so let me show you a couple pictures, and we will be right back. Okay, so these are just a couple pictures that I usually draw on the board with my kids. So I thought we could take a look at these and see if they help out at all. All right, so here we go. We have DNA replication. So obviously we have one DNA molecule is going to become two, and I don't have my pointer ability here like I did in the PowerPoint, but you can see how you have sister chromatids being made, and they're half old, half new. That's semi-conservative, right? Next thing I tell my students to know is, okay, definitely make sure that you understand the structure of a nucleotide, right? And this is a monomer of a nucleic acid. You have that phosphate group, okay? You have deoxyribose is the sugar. It's a pentose sugar, 5 carbon. And then you have your nitrogenous bases here. In DNA, we have A, T, C, and G. Okay. And yeah, you could dive into this stuff. As you know, I can't stand memorization, but there are some times when you just got to do it. You just got to put your head into it and memorize facts, you know. And a good example of that is here with your pyrimidines and purines and things like that. So the longer the word pyrimidine, it's actually just one ring, and purines, the shorter word, has 2 rings. Okay, and here you can see an adenine bonded to a thymine, and there's 2 hydrogen bonds in between them. Okay, and notice how it's antiparallel, so the 5' goes up here. That means at the bottom, okay, there it is, it actually comes off of the bottom there. So if we could see it all at once. And we call this antiparallel because now on the other side you have the 3' end, okay, conversing the 5' end, and down here at the bottom then we'd have the 5' end, and notice it's upside down there. Okay, so once you know that, we talked today about the origin of replication, how you have certain spots where DNA replication can begin, and it opens up these bubbles, and then at either side you're going to have the replication fork. So of course you need an RNA primer, and you're always going to build in the 5' to 3' direction. At this replication fork, you should know what all these enzymes do. Topoisomerase breaks and reforms so you don't overwind your double helix. Helicase unzips, single-stranded binding proteins come in here. And now very early on, if this is the origin of replication here, we have primase coming in and laying down that primer. Now that we have a primer laid down, now DNA polymerase says, just like with those Lego blocks, oh, I know what to do. And he starts building the, maybe it's a sheet, I don't know. But he starts building the nucleotides, right, in the 5' to 3' direction. Okay. And let's speed it up a little bit more. Here you can see DNA polymerase 3 still doing its job. Okay, but now you can see a whole lot of little fragments here, the Okazaki fragments on the lagging strands. Right? So you can see how DNA was built there. And again, you can pause this or think about it as we keep going here. And these multiple bubbles, multiple origins of replication really speed this up. And now DNA polymerase 1 comes in here and says, oh, let me help you here. And he swaps out the RNA for DNA. Right? And then what's going to happen next is DNA ligase says, oh, look at that. All the RNA is gone, but we have a gap here. Because remember, I told you DNA polymerase isn't necessarily the smartest enzyme. So here's the next problem with it. Yes, it knows how to elongate. It doesn't know how to start from scratch, but it knows how to elongate nucleotides. But the thing is, when you're building and you get that close, it doesn't know how to make this final bond here. Okay, it doesn't know how to connect them. So that's why we need ligase to come in, right? So DNA ligase is going to come in and connect those Okazaki fragments for us. Okay. All right. Well, again, that was DNA replication, and I hope you found this helpful. Good luck. Take care.